Hi, good morning, everyone. I hope you had a good weekend, had a nice evening in the yesterday. So let's start today with the glycolysis, citric acid cycle, metabolism. All the biochemistry's important cycles. We will start now. I hope students will join in. 3. So as we know, let's start with the glycolysis first. So our we have discussed till now carbohydrates, fatty acids, lipids, proteins, amino acids, water. Yeah. So we have discussed quite number of our basic molecules in biochemistry. Then we have discussed also the medicinal diseases that would happen in a body, like jaundice, liver diseases, and also we have discussed about um, hyperthyroidism due to the uh, change in the thyroid level of hormones. So today we're gonna discuss about various amino acids, these all biomolecules, how they are being used in our cycle, how they're producing energy, and how essential they are in a body. So we're gonna take one part one by one related to that. So this will be not in the PPT form. I strictly don't like this topic to be in PPT. So I wanted to go through this in WordPad thing because this is very extensive, and I will go word by word so that we can understand each aspect of it in easy manner. So most organisms that oxidize carbohydrates as their primary source of cellular energy. So glucose is the most common carbohydrate. So we have discussed that glucose is an instant source of energy to produce energy in the microorganisms. So basically they're using two methods, that is your cellular respiration, yeah, cellular respiration, and and fermentation. And there's another third method that is called aerobic respiration, which is another method in which the final electron receptor is an anorganic substance than the oxygen. So let's talk first aerobic respiration. That is your catabolism or oxidation of carbohydrates. That is your oxidation or aerobic respiration of your carbohydrates. So it's basically three methods are being taken care in glycolysis. In the aerobic respiration of a carbohydrate. The first is your glycolysis, then comes your Krebs cycle, then comes your electron transport chain. These three cycles are mainly playing the essential role in it. So how glycolysis is playing? So in glycolysis, what is really happening is your oxidation of glucose to pyruvic acid with the production of some ATP and energy containing NADH. So yeah, as the name says, lysis. So something is being broken up here. So your glucose is broken up by oxidation, glycolysis, to pyruvic acid. At the end, you have a pyruvic acid being produced, and there is also some energy is produced like ATP and also NADH. So in the form of ATP and NADH, your energies are produced in the glycolysis from glucose, and the end product that is produced is your pyruvic acid. Now comes second part is your Krebs cycle. So once your glycolysis is over, you have your pyruvic acid, then it will continue further. So in this case, you will have a derivative of a pyruvic acid, that is your acetyl coenzyme A. Acetyl will convert to carbon dioxide with the production of some ATP, energy containing NADH, another reduced electron carrier FADH2. So in this case, after the oxidation of this derivative of pyruvic acid, it will produce CO2, ATP, NADH, uh, reduced electron carrier, and FADH2. So all these things being are produced in it. Then comes your electron transport chain, that is NADH and FADH2 that have been produced now, they will get oxidized. What is happening in each part, like from glycolysis to Krebs cycle, electron transport chain, the end product that are being produced, they are being utilized and getting oxidized. So like here we have produced enough of your NADH and FADH2, and they will get oxidized and they will contribute to the electrons. And they have carried from the substrate to cascade of oxidation reduction reactions involving a series of additional electron carriers. So energy from these reactions is used to generate considerable amount of ATP, and in respiration most of the ATP generated in the steps. So mostly it is used in the respiration case. So yeah, so these were the 3 steps, students: glycolysis, Krebs cycle, electron transport chain. Now we will continue one by one each pathway we will take, and to understand them what is really happening in them. So glycolysis, also called as Embedon-Marov pathway. So each terms that are coming here, students, in this word, it's very important. It's very important from the examination point of view. It's very important from MCQs. If you're preparing for any entrance exam, so most of the questions from a biochemistry is being asked from these cycles only. So be ready with that. So as we know, Greek word glykis means sweet. Yeah, lysis means to break. So something sweet is being broken up. That is your glucose. So molecule of glucose is degraded into enzyme catalyzed reactions. So lots of enzyme are used in this to yield 2 molecules of 3 carbon compound pyruvate. So pyruvate as glucose is a 6 carbon atom, and it will produce 2 molecules actually of 3 carbon atoms called as pyruvate. And also during this time, NADP plus is reduced to NADH, and there is net production of 2 ATP. So students, please remember these exact terms. How many molecules are being produced? 2 molecules of a pyruvate, yeah, NADPS is reduced to NADH, and 2 ATP molecules are produced. So glycolysis does not require oxygen and can occur whether present or not. So it's independent of oxygen. Whether oxygen is present or not, it will keep doing its function. Yeah. So that's basically the reaction of your glycolysis. You have a glucose, 2 NAD plus molecules, 2 ADP molecules, 2 phosphate molecules. And then you produce 2 pyruvate, 2 NADH, 2 molecules of hydrogen, 2 molecules of ATP, and 2 molecules of H2O2. So that's the net overall reaction of your glycolysis. Here 2 molecules of ATP were needed to glycolysis started, and 4 molecules of ATP are generated. Yeah, and there is net gain of 2 molecules of ATP for each molecule of glucose that is oxidized. So let's see one by one with various enzymes, as it's important aspect to remember them forever. So it's very important in exam. It comes always this glycolysis cycle, so you need to remember it by heart actually. So you don't have to remember these structures, that will be too much actually, but just remember the names at least. So first is glucose. So glucose, in the presence of first ATP, it will produce ADP. Yeah. So it will take one phosphate group from it, and then it will form glucose 6-phosphate. Yeah. And during this reaction, your first enzyme that is used is your hexokinase. Yeah. The enzyme that is used is known as hexokinase. Then after glucose 6-phosphate, it will produce fructose 6-phosphate. So we can see glucose to 6 is something change in the structure is happening, isomerization is happening. So in that case, enzyme that we use is phosphohexose isomerase. Phosphohexose isomerase. Then from fructose 6 phosphate, again ATP is being used to produce ADP. We will take one phosphate group, and this is second priming reaction. So it will produce fructose 1,6 biphosphate. So we are adding one more phosphate to it. So the enzyme that we will be using here, fructose phosphofructose kinase 1, because kinase are used whenever we have to add one phosphate group to our substrate. So we use kinase whenever we want to add one phosphate group, and we use phosphatase whenever we want to remove one phosphate group. Yeah, isomerase to change the reaction. Yeah, hexokinase, phosphofructokinase, and then further from fructose 1,6 biphosphate, the cleavage of 6 carbon sugar phosphate to 2,3 carbon sugar phosphate group. So it will be broken up into two different molecules now. That is glyceraldehyde 3 phosphate and dihydroxyacetone phosphate in the presence of aldolase. So aldolase work is to break down your two molecules into two parts. So from here we can see these cycles. So the blue part is separated here and the red part is being separated here. So you have glyceraldehyde 3 phosphate. And then further the last enzyme will be used here after the fifth enzyme is triose phosphate isomerase. Triose phosphoisomerase. So this will, what it will do, it will make glyceraldehyde 3 phosphate molecules into two molecules. It will produce two molecules of the same group. So this first part is known as your preparatory phase in this blue one, and it's a conversion to the glyceraldehyde 3 phosphate. So in this case, what we have discussed so far is your glucose is converted into glyceraldehyde 3 phosphate. That's it in the preparatory phase with the help of these five enzymes: hexokinase, phosphohexoisomerase, phosphofructokinase, aldolase, triose phosphate isomerase, and so on. Yeah, the first enzyme, and then your first product is glucose 6 phosphate, fructose 6 phosphate, fructose 1,6 biphosphate, glyceraldehyde 3 phosphate, and then two molecules of the same. Now second phase of your glycolysis that we have to learn about is your payoff phase, which is oxidative conversion of your glyceraldehyde 3 phosphate to pyruvate and the coupled formation of ATP and NADH. So now ATP and NADH production will be taken care of, and you will have end product as a pyruvate here, two molecules of pyruvate. So now what will happen? So first oxidation and phosphorylation reaction will happen. So NAD plus will be reduced within the presence of two phosphate groups, so it will produce 2 NADH. And from the glyceraldehyde 3 phosphate we will have 1,3 biphosphoglycerate. And then afterwards first ATP forming reaction, so this will be happen in the presence of glyceraldehyde 3 phosphate dehydrogenase. Then in the presence of phosphoglycerate kinase, again ADP molecules will produce 2 ATP molecules to produce 3 phosphoglycerate. Yeah. And then in the presence of phosphoglycerate mutation will happen to produce 2 phosphoglycerate. And then water molecule will be released with the help of enolase enzyme and phosphophenol pyruvate. After the phosphophenol pyruvate production is taken care, and then you will have your again ADP is producing 2 ATPs, and then you will have in the presence of pyruvate kinase pyruvate at the end. You have a production of your pyruvate at the end. So in this way this whole glycolysis cycle works out. Yeah. So so we have to see that the first two molecules of ATP are producing, giving rise to 2 ADP molecules. Yeah. And then at the end 2 ADP molecule here, 2 ADP molecule here, like 4 ADP molecules are being used here, and then 4 ATP is being produced. So 4 ADP, 2 ATP, on the left hand side, and then 2 ATP on the left hand side. So if we subtract them, so we will have something like 2 ATP were needed to glycolysis started, and 4 molecules of ATP are generated by the process. So there is net gain of 2 molecules of ATP. So net gain that is there will be at the end is 2 ATP, and each molecule that is oxidized. Yeah. So this is the overall reaction. Glucose plus two NAD plus 2 ADP plus 2 phosphate plus 2 pyruvate plus NADH, 2H plus 2 ATP and 2 hydrogen molecule, and so on. Okay. I hope there is all is clear in this case. There is no question. Shall we continue further? So that was the basics of glycolysis here. So based on that, we continue further to your Krebs cycle. So there are some alternatives to glycolysis, that is your pentose phosphate pathway and enter-Dudov pathway. Dudov pathway. Yeah. So in bacteria, in addition to the glycolysis for the oxidation of glucose, most common that the bacteria that are using are pentose phosphate pathway and enter-Dudov pathway. So in pentose phosphate pathway, that is your hexose monophosphate shunt, HMS. So this provides the breakdown of 5 carbon pentose sugar as well as the glucose. So basically 5 carbon sugars are being broken up as well as the glucose. So key feature that it produces important intermediates used in the synthesis of nucleic acids, and glucose from carbon dioxide in photosynthesis and certain amino acids. So the pathway is an important producer of reduced coenzyme NADH from NAD+. So this pathway yields net gain is 1 molecule of ATP from each molecule of glucose oxidized. So bacteria that use this pathway includes Bacillus subtilis, E. coli, Lactobacillus mastodoridis, and Enterococcus faecalis. And in the enter-Dudov pathway, for each molecule glucose, this pathway produces 2 molecules of NADH in this case and 1 molecule of ATP. And for use in the cellular biosynthetic reaction, and in this pathway they are found in the mostly in the gram-negative bacteria. So including Rhizobium, Pseudomonas, Agrobacterium, and so on, and not found in the gram-positive bacteria. So these were pathways for basically for the bacteria, not in the human being. In the human being, the first pathway is taken care. So as the aerobic, the broken down of glucose to pyruvic acid is done, then further this glucose is channeled, this pyruvic acid is channeled into the next step, that is known as fermentation or cellular respiration. Fermentation or cellular respiration. So in cellular respiration, it is defined in which ATP is produced for sure, molecules are oxidized, and finally the electron receptor are inorganic molecules. So there are two types of respiration that occurs, that is aerobic and anaerobic. In an aerobic, the final electron receptor is oxygen. In anaerobic respiration, it is an organic molecule. That's the main difference between aerobic and anaerobic, that in anaerobic the final electron receptor is oxygen, and in the anaerobic respiration it is an inorganic molecule other than oxygen. So let's discuss Krebs cycle, citric acid cycle, or tricarboxylic acid cycle. So the pyruvate produced by glycolysis is oxidized completely, generating additional ATP and NADH in the citric acid cycle and by oxidative phosphorylation. However, this can occur only in the presence of oxygen, and oxygen is quite toxic to organisms that are obligate anaerobes and are not required by the facultative anaerobic organisms. So, in the absence of oxygen, one of the fermentation reactions takes place in them to regenerate NAD plus lactic acid fermentation is one of these pathways. So, in this eukaryotic cycle, the citric acid cycle occurs. Yeah? So, we're going to discuss about now citric acid cycle, the next cycle, the Krebs cycle or TCA cycle, tricarboxylic acid cycle. And this is happening in the matrix of the mitochondria, and bacteria uses this TCA cycle to generate energy, but they don't have mitochondria, so reactions takes place in the cytosol with the proton gradient for the ATP production. So let's discuss this very nice citric acid cycle here. So we have, first of all, to initiate our cycle, we have this citrate. Remember, we have this molecule called citrate, and this citrate will produce isocitrate with the help of enzyme aconitase. Yeah, and then once the isocitrate are produced, then you will have your alpha ketoglycerate. After the in the presence of isocitrate dehydrogenase. So here also NAD plus is reduced to give your NADH, NH plus, and carbon dioxide is released. Then further from alpha ketoglycerate, your succinyl coenzyme is being produced in the presence of alpha ketoglycerate dehydrogenase, and in the presence of your NAD plus and coenzyme A, and will again produce NAD plus carbon dioxide. So once this is done, the succinyl coenzyme A is being produced, then it will produce succinate in the presence of succinyl coenzyme synthetase, where your GDP, guanine diphosphate plus guanine diphosphate plus phosphate give rise to, it will remove this coenzyme A and also the GTP molecule. So from this succinate, then further succinate dehydrogenase, we will have a fumarate, and then from fumarate, with addition of water and the presence of fumarase enzyme, we have a malate. Then again NAD plus is being 3 times being reduced. In the presence of malate dehydrogenase, it will produce oxaloacetate, OAA. Then in the presence of water with the citrate synthesis, it will produce citrate. So this cycle, your citric acid cycle, from citrate, isocitrate, alpha ketoglycerate, succinyl coenzyme A, succinate, fumarate, malate, oxaloacetate, with the help of these enzymes and the NAD plus being reduced each time, and also GDP is being reduced to give GDP, it will make this cycle to be a cyclic. This will be your citric acid cycle. On the other side, as we know, we are having this pyruvate, from the glycolysis cycle. From the pyruvate, in the presence of pyruvate dehydrogenase, again your NAD plus is being reduced to NADH plus. In the presence of coenzyme A, you will have a acyl coenzyme A. Yeah, you will have a acyl coenzyme A, and this could acyl coenzyme A will help with the help of oxaloacetate to produce your citrate by removal of this coenzyme A. Yeah. And further from this pyruvate, we can have this reaction in the presence of pyruvate carboxylase to produce oxaloacetate. Yeah, HOC3 minus plus ATP give rise to ADP, and your OA is being produced. So here this cyclic, another cyclic cycle is being taken place here too. Yeah, I will repeat this cycle. So these cycles are even though they are a bit complex, but once we understand the basic concept behind them, and then we have to draw them, students. Once whatever the cycles that I'm telling you today, so please do not make these assignments on the computer. Do it with your own hands. It should be handwritten. because if you do these cycles in your hand, you will remember them forever. Yeah, so it will be good for your remembrance actually. So please do make these assignments as handwritten. So I will repeat this cycle. So you have a pyruvate being produced from the glycolysis cycle, and then from there you have your sterile coenzyme A, and then in the presence of citrate synthase and oxaloacetate produce citrate, and beyond that pyruvate could also produce oxaloacetate in the presence of HOC3 plus ATP, giving rise to ADP and pyruvate carboxylase enzyme. Uh, and then citrate is producing isocitrate in the presence of aconitase. Then isocitrate is producing alpha ketoglutarate in the presence of isocitrate dehydrogenase in the presence of NAD plus and NADH. Then alpha ketoglutarate is producing succinyl coenzyme in the presence of alpha ketoglutarate and dehydrogenase. And then succinyl coenzyme is producing succinate in the presence of succinyl synthetase, and then succinate is producing fumarate in the presence of succinate dehydrogenase, and fumarate is producing malate in the presence of fumarase, and malate is producing oxaloacetate in the presence of malate dehydrogenase, and then oxaloacetate is producing citrate in the presence of citrate synthase. And wherever your NAD plus is being reduced, first step is here, 2, 3 and 4. Yeah, so there are 4 steps where NAD plus is being reduced, and there's GDP is also being reduced here in the presence of succinyl coenzyme to succinate also. So remember these parts. We should not remember these structures. It's not required. It will be too much. But for your knowledge, you can remember them. No worries. So that's it about the TCA cycle. That was the main behind part of these cycles. So I will share these. Okay. Now we take a little bit about electron transport chain. So because in this, so we have discussed glycolysis cycle, we have discussed the Krebs cycle in the aerobic respiration of carbohydrate, and the third part is your electron transport chain. Yeah. So we will discuss about this now. Just a second, coming back. Okay. So this is the last part of electron transport chain. So basically, there are many complexes here that are playing role, and these complexes play together very essential role, and together they function in a very nice manner. Only then we can see the results, right? So in the electron transport chain, it consists of sequence of carrier molecules that are capable of oxidation and reduction. So in this basically many carrier molecules will be present which will do your oxidation and reduction, and as electrons are passed through the chains there is a stepwise release of energy and used to be called drive through the generation of ATP. So in eukaryotic cells it is contained in the inner membrane of mitochondria, and the prokaryotic cells it is found in the plasma membrane, right? So here we having these we can see these very nice diagram here where we have a NADH dehydrogenase complex, then we have a cytochrome BC1 complex, and then we have a cytochrome oxidase complex. Yeah, so these all complexes they having various molecules which will produce our ATP at the end. Yeah, so basically here we can see is in the first step uh, NADH is being oxidized to produce NAD plus, giving rise to FMN. Then FMN will produce two molecules of hydrogen, and beyond that it will keep producing your Q. We'll discuss about that complex also. So here we have three classes of carrier molecules that are involved: flavoproteins, cytochromes, and coenzyme or ubiquinins. So these are your flavoproteins, that is FMN contains flavin, and they are coenzyme that are derived from the riboflavin vitamin B2. And one of the important coenzyme is your flavin mononucleotide. Flavin mononucleotide. Then comes your cytochrome complex. So they are the proteins with containing iron group, and they alternatively they are produced in the reduced form of iron or oxidized form of iron. So the reduced form is Fe2 plus and oxidized form is Fe3 plus. So the cytochromes includes your cytochrome B, C1, A, and A3. Cytochrome B, C1, C, and A3. And ubiquinons or coenzymes, these are small non-protein carriers. Yeah, remember these three different classes. That is your flavoproteins, then comes your cytochromes, then comes your coenzymes or ubiquinons, which carry your protein further. So in the electron transport chain of bacteria is somewhat diverse. The particle carriers are ordered in which the functions may differ. So much is known about the electron transport chain in the eukaryotic cells till now, but not in the prokaryotic cells. So first case, what the first step is happening is transfer of high energy from NADH to FMN is taken place, and this involves the passage of hydrogen atoms with 2 electron to FMN, which then picked the additional hydrogen ions from the surrounding aqueous medium, and NADH is oxidized to NAD plus and FMN are reduced to FMNH2. So here we are having these NADH given rise to NADH, and this FMN is being produced here, and this producing two hydrogen molecules, and also this FMN is further reduced to FMNH2. Yeah. And then FMNH2 passes two hydrogen molecules to other side of mitochondrial membrane to pass 2 electrons to Q. Yeah. As a result, FMNH is oxidized to FMN, and Q picks an additional 2 hydrogen molecules from the medium and release to the outside the other membrane. So this Q will also release two molecules of hydrogen from it after taking in from the FMN and taking 2 hydrogen molecules from the inside membrane and producing to the outside membrane of your mitochondria. Then they are successfully passed to the cytochrome B, cytochrome C1, cytochrome C, then cytochrome A and cytochrome A3. Yeah, all these different cytochromes, it will be transferred to them. Cytochrome B, cytochrome C1, cytochrome C, cytochrome A, then cytochrome A3. So electrons are being transferred like in this cytochrome complex in the next step. So each cytochrome is the chain is reduced to picks up electron and oxidize, it gives up electron, and the last cytochrome A3 carries electrons to the molecular oxygen, which becomes negatively charged and then picks up protons and medium to the water molecule. So this will add 2H plus half oxygen, giving rise to water molecule here, right? And beyond that there will be six actually hydrogen molecules are being produced, 2 from FMN, 2 from Q and 2 from cytochrome B, a cytochrome complex. So total 6 hydrogen molecules, right? So these 6 hydrogen molecules and water molecule here, in that third step, in the presence of ADP and 3-phosphate group, it will produce your ATP in the presence of enzyme ATP synthetase. Yeah, so that's how the third step is so important. So FADH adds 2 electron to the electron transport chain at the lower level of NADH. Because of this, electron transport chain produces 1/3 less energy for ATP. And FMN and Q accept release protons as well as electrons are the carriers. And this electron flow down this thing is called as elective transport of protons. So chemoosmotic mechanism of ATP generation. So in this, what is really happening is that is a mechanism of ATP synthesis using the electron transport chain. This method is known as the mechanism of ATP synthesis using the electron transport chain is called chemoosmosis. Just a second. So the summary, I want to come to this point actually, summary of aerobic respiration that we have just discussed. That is, your electron transport regenerates NAD plus and FAD plus, which can be used again and again in glycolysis and Krebs cycles. Yeah, and various electron transport in the electron transport chain generates around 34 molecules of ATP. For each molecule, glucose being oxidized, producing 10 NADH and 2 FADH2. And total 38 molecules can be of ATPs generated from 1 molecule of glucose in prokaryotes. You can see so much of energy is being produced in prokaryotes. And total 36 molecules of ATP produced in the eukaryotes from 1 molecule of glucose. But some energy is lost during the electron transport shuffling. Yeah. So here we can see glucose is producing 2 molecules of your pyruvic acid, and pyruvic acid is producing with the help of Krebs cycle, acetyl coenzyme A, and this Krebs cycle is happening in the reversible cycle, and then from this Krebs cycle you are having 38 molecules of ATPs produced. So how these all reactions are happening? If we see the right hand side, 2 ATP molecules are produced from the glucose, and then 2 carbon dioxide, 4 molecules of carbon dioxide here, 6 total, and then further 2 molecules of ATP is produced from the Krebs cycle. So 2 plus 2, 4. And beyond that, this electron transport chain that we have discussed, so I need 2 molecules of NADH, 2 molecules of NADH, 6 molecules of NADH, 2 molecules of FADH. So total 10 NADH plus 2 FADH. So they will take this electron with the help of, I have discussed, electron transport chain and chemoosmosis, and they will produce 34 molecules of our ATP. Then this 34 plus these 4 molecules of ATP will produce 38 molecules of ATP. So that's how your all carbohydrate cycle, the aerobic respiration of carbohydrate in these 3 different cycles will produce this much amount of ATP at the end. So that's a summary of all the things that we have discussed today in a nutshell. Yeah. So go through it. So somebody asks you in exam, write down about the glycolysis cycle or citric acid cycle or electron transport chain, or explain the summary of all glycolysis, citric acid cycle and electron transport chain, how much is energy is produced. So you have to explain that diagram. So this is the second part in that series. So this was actually, I can repeat this, I should have started with this lecture actually, just explaining that we have also discussed an enzyme reaction and how things are working, just to have your clear fundas about various terms. Sometimes we tend to forget them. Metabolism, which is a sum of your both catabolism and anabolism, in which energy is released. We have a balancing act in the body. So catabolism is release energy, anabolism is require energy. So catabolism is a enzyme regulated chemical reactions that releases energy, whereas anabolism is an enzyme regulated energy requiring reaction. Yeah. So these reactions are very necessary for the growth, and the coupling of these reactions is able to produce energy in the form of adenosine triphosphate. So what is adenosine triphosphate? We have one adenosine as an your nitrogenous base pair, ribose sugar, and 3 phosphate groups. So if we break this bond, then a lot of energy is being produced. Somehow if we break this, some energy is produced. If we break this, little bit energy is being produced. So in this way our cycle is being produced. So these reactions generates the material for growth, and this coupling of energy requiring energy releasing reactions is made possible through the molecules adenosine triphosphate. And ATP stores energy derived from catabolic reactions and releases later to the anabolic reactions and perform other cellular work. So ATP is an adenine, ribose and 3 phosphate group. So when terminal phosphate group is split to ADP is formed, and energy is released to drive anabolic reactions. So ATP is producing ADP plus phosphate plus energy is released. And then this energy could be also be resynthesized in the presence of again reversible reaction, and that will be known as anabolic, but catabolic will be the ATP synthesis. So enzyme, we have discussed that they act as a catalyst, biocatalyst, in which there is a substrate and they get attached to it to produce your product. So there is a consist of protein portion called apoenzyme and non-protein portion cofactor, and this cofactor could be zinc, iron, magnesium, and it could be also organic molecules. So then together apoenzyme and cofactor they form holoenzyme, which is an active enzyme which will work for it. Further energy production is done in the formation of oxidation reduction reaction. Oxidation is basically removal of electrons from an atom or molecule, a reaction that often produces energy. And reduction is the addition of one molecular electron to an atom or molecule. Right, generation of ATP. So most of the reactions that we have just discussed now during this during the oxidation reduction reactions, they are trapped within the cell by formation of ATP. So phosphate group is added to ADP with input of energy to form ATP. So addition of phosphate to chemical compound is phosphorylation. An organism use three mechanisms of phosphorylation to generate ATP from ADP. Substrate level of phosphorylation, so ATP is generated when a high energy phosphate is directly transferred from phosphorylated compound to ADP. So this is substrate level phosphorylation. You have many CCC with phosphate plus ADP giving rise to ATP. So this from this carbon chain your ATP is produced. Then oxidative phosphorylation, just we had just discussed, where electrons are transferred from the organic compounds to another electron group, usually from like NAD plus and FAD plus and so on. So this is just giving a reference of all reactions, how things are working there. So I think we should be done here. Tomorrow we will discuss about lipid metabolism, right? And we will discuss. We will start with another chapters where related to medicinal biochemistry tomorrow. So I think we will be done in this week by Friday. Yeah. So yeah, we will have classes till Friday, I guess. So in the next four classes, we will go in more detail about various aspects of biochemistry, and discuss things. So today was the day for this, mainly the cycles. There is one lipid is left. Maybe I want to do it today, but this will be too much for all of you to take it one day. So basically, it's also the same, but here the lipid metabolism is taking place, like protein and amino acids catabolism. It's also of 3 or 4 cycles, not much. We can do this also today so that we are happily done with this part also. Yeah. We started quite late, so we can do it. So protein and amino acid catabolism. So some bacteria and fungi, particularly pathogenic food spoilage and soil microorganisms, can use proteins as a source of carbon energy. So like protease, that are enzymes that break down proteins into amino acids, and amino acids are first deaminated, then enter to the Krebs cycle. So basically intact proteins cannot cross bacterial plasma membrane, so bacteria must produce extracellular enzymes called proteases and peptidases that break down the protein into amino acids which can enter the cell. So many of these amino acids are used in the building bacterial proteins, but some could be used for the breakdown of energy. So it is the way amino acids are used that they broke down into some that can enter the Krebs cycle. So in this reaction, so basically for your amino acids, so following reactions are taking place. First is deamination or transamination. So in this deamination or transamination, so amino group is removed from or transferred or converted to ammonium ion and then excreted, and the remaining organic acid can enter to the Krebs cycle. Then comes your decarboxylation, that is a carboxylic group is removed, and dehydrogenation, a hydrogen is removed from it. Yeah. So you are having this glutamic acid in the presence of oxaloacetic acid transamination will happen. So you will have transamination means a amine group is changed. From this amine group is here in this oxaloacetic, it will make a aspartic acid. It will remove the oxygen and will attach as a amino group, and it will itself become a alpha ketoglutaric acid. It will form a double oxygen bond here. Yeah, so that's how the reactions will change. So that was the then transamination, how this whole reaction is taking place. So you're having carbohydrates that we have just discussed. They will produce sugars. Then in the glycolysis, glyceraldehyde 3 phosphate was produced. Then pyruvic acid. Then acetyl coenzyme A. It went into the Krebs cycle, and then it produced electrons. And with the help of electron transport chain, it produced water molecules and lot of ATPs. And beyond that, all these reactions are attached to your lipids and proteins also. So here lipids are also producing glycerol and fatty acids. So glycerol could produce glyceraldehyde 3 phosphate here, and your fatty acids could produce acetyl coenzyme A. So they come and be part of the same reaction. And beyond that, your proteins could come also play very important role. Amino acids could produce here also some proteins here in this glycolysis cycle. Acetyl coenzyme A also in the Krebs cycle. So all in all, your transamination, your protein catabolism, your lipid catabolism, your carbohydrate metabolism, they are all interconnected with each other and helping out in the reaction. So that is the very broad picture of protein and amino acid and carbohydrates catabolism. Now second part is a lipid catabolism. So in this, microorganisms frequently use lipids as energy source, and the triglycerides or triacylglycerols. So esters of glycerols and fatty acids are common energy source in this case. So in the first step, lipases are enzymes that break down the fats into fatty acids and glycerol components, and beta oxidation is the breakdown of the fatty acid into two carbon segment, that is a acetyl coenzyme A, which could be used further in the Krebs cycles. So functions of lipids in microbes, so they are very essential for the structure of membrane. Reserves energy, could be source of carbon. 90% of lipid is triacyl glycerol, and triacyl glycerol produces glycerol plus 3 fatty acids. And major fatty acids metabolism is your beta oxidation. So lipids are broken down into the constituents of glycerol and fatty acids. So glycerol is oxidized by glycolysis and TCA cycle. So what is happening in this actually? So your lipids of fats, they are producing glycerol or fatty acids either. So beta oxidation will happen, producing a acetyl coenzyme A, which could be used in the Krebs cycle. Or on another side, glycerol will produce dihydroxyacetone phosphate, which will produce a glyceraldehyde 3 phosphate, which could be used in the glycolysis cycle to produce pyruvic acid and hence acetyl coenzyme A. So that's basically the lipids working off as a beta oxidation pathway takes place. Yeah, that's a summary of your lipids as a beta oxidation pathway here. So this we have discussed a bit summary today, students. We will later discuss about in detail tomorrow. Yeah, so I want to stop this part here today only, and we will do this tomorrow, this part, because this is a bit vast now. This will be too much to take in. I have shared the reactions also of this. So that's it for today's